Okay, I hope I am audible, and welcome to Let's Crack NEET UG English. We are still continuing with the cell division part. In the previous lecture, we talked about all the stages of Meiosis I. Meiosis I is a lengthy process, it's a long process. And in Meiosis I, we also saw that there were, in prophase, there were 5 subphases like leptotene, zygotene, and so on. So we talked about those stages also. What would happen if Meiosis I is completed? After that, the 2 daughter cells have to divide one more time. Because in the beginning we said in meiosis, the DNA replication takes place only once, but the cell has to divide 2 times. So all those things we have already talked of. We'll continue with meiosis II. Let us quickly go over our basic information. This lecture which I am taking right now is on the channel Let's Crack NEET UG English. Very soon I'm going to be starting lectures on Biology 101. I'm already taking lectures on Let's Crack NEET UG. And very soon the special classes are also going to start. So maybe in a day or two, I'm going to put up all the schedules, all complete schedules, like on every channel, the dates, the topic or the subtopic, so that you know exactly which chapter is being covered on which channel, and you will be able to plan your schedule accordingly. One more important thing which I have to tell you right now is about the payment plan, but before that, don't forget to install this app so that you get all the benefits of it, live lectures and unlimited resource and everything. This is the main thing which I want to talk about today, is because very soon this fee structure is going to change. It is going to increase. So it is better that you take a plan so that you get the advantage of the existing price. Very soon this price range is going to totally change. And if you take this decision right now, don't forget to use this code so that you also avail the 10% discount based on this code. We have had almost 4, 5, 6 lectures by now. So this much time is more than enough to understand how a teacher takes care of a particular topic, how easy or difficult that topic is for you. And if you think this is really helping you, then you need to take this call immediately before the plan changes, or before these the prices they change or they are hiked, because this is going to increase. So take advantage of this lower pricing plus the discount which you can avail with the code, that is Neela. Prophase II is what we are starting with. But before that, let us quickly go over some basic points which we talked of in the earlier lectures. In Prophase I, there were 5 subphases, that is leptotene, zygotene, pectine, diplotene, and dikinesis. After that, the cell goes into metaphase I. What happened in metaphase I? In metaphase I, the chromosomes, they got aligned on the equatorial plane, but they were aligned in a pair. And the spindle fibers which were attached, they were attached in a manner that from one aster only one chromosomal fiber was coming, which was getting attached to only one chromosome of the pair, and from the other end other chromosomal fibers were attached. So if we have to just enlarge this thing, this is one chromosome and this is the other one. This is one pair. The spindle fibers are arranged like this. In case of metaphase, we said that only one chromosome is on the equatorial plane, and to the centromere the fibers were getting attached from both the poles. But here one chromosome of the pair is attached to the fiber coming from one pole, and the other chromosome of the pair is attached to the fiber coming from the other pole. What will happen in anaphase 1? Were you able to figure out? If this is the cell, then when these spindle fibers contract, one chromosome of the pair goes towards one pole and the other chromosome of the pair goes towards the other pole. So the two chromosomes of the pair, they get separated. This is in anaphase 1. Will there be interzonal fibers? In case of anaphase 1, will there be interzonal fibers? This was given to you as homework. So in anaphase, no interzonal fibers, as the centromere is not splitting, as centromere does not split. So now when this cell goes into telophase 1, what will happen in telophase 1 is the chromosomes would have reached the opposite poles, the nuclear membrane will assemble and two nuclei will be formed. Now how many chromosomes have reached each pole? So here there are 2 and here there are 2. This will be followed by cytokinesis 1. Sometimes may not take place, this cytokinesis 1, but if cytokinesis 1 takes place, we would have 2 daughter cells, each having 2 chromosomes in its nucleus. We started with a parent cell having 4 chromosomes. So after meiosis 1, each daughter cell has half the number of chromosomes as that of the parent cell. Okay, so I guess all those things which were given to you to be done as homework, you got them right. And if you did, that means you have understood everything clearly. And suppose some of you got few things wrong, so what you need to do is you go back to the original complete lecture, revise it so that whatever that missed out thing was, that concept also gets cleared. And once we finish meiosis II and everything, moving forward, we will also plan a quiz so that you are able to answer the question. So you prepare this complete chapter, cell cycle, cell division, all the types of division, and then maybe we'll have some doubt solving classes and a quiz session also for you. So cytokinesis 1 is done, we are going theoretically. So we assume that cytokinesis 1 takes place, and now the cell enters meiosis II. In meiosis II, what we have to do is, we have to do or draw all the changes in both the cells. Because whatever is happening in one cell will happen in the other cell also. So what we do is, we keep both the cells together, we draw the changes in one cell, but we keep telling ourselves that everything what is happening in one cell is happening in the other cell also. So now the cell is in prophase 2, and here in prophase 2 we don't have any subphases. Do you think we need to draw early and late? Now we know what happens in early stage. In early stage, nuclear membrane dissociates, nucleolus dissociates, all those changes. So we can straight away draw the final stage. And let us not forget to draw both the stages, both the cells together. These are the two daughter cells which have entered meiosis to prophase 2. I'm going to draw the daughter cell here so that the change also becomes clear. Daughter cells. These daughter cells are formed as a result of meiosis 1, and each daughter cell has only one set of chromosomes. This has one set and this has one set. It has its nuclear membrane reassembled now. Here also the nuclear membrane is proper and the cell division has also taken place. And suppose we are talking about the animal cell. So there was a centriole here and a centriole here. Now both the cells which are formed as a result of meiosis 1 have entered prophase 2. So what will happen? Again the cell has to divide. So now the centriole splits, nuclear membrane dissociates, nucleolus dissociates, organelle dissociate, chromatin fibers condense, all those changes are taking place. We will write down those changes also. But the most important thing is that the centriole which was at a particular position, where were the centrioles, or let me ask you the same thing in an other manner. Where were the poles? If you look at the cell, there was one pole at the upper end, the other pole was at the lower end. Now, the orientation of the poles is going to be at right angles to the first orientation. So first orientation is vertical, now second orientation is going to be horizontal. That means one centriole will come here, the other will come here. Same thing will happen in the other cell also. We will draw one or two stages where both the cells we can take, and then we can leave one cell because by that time we'll know that exactly same thing is happening in the second cell also. Spindle fibers would also appear, aster rays will also appear because this is prophase complete, prophase 2. Here also the aster rays, and now the spindle fibers. So spindle fibers are going to be the continuous spindle fibers, discontinuous spindle fibers. In this case also the second cell also. So is the orientation of the spindle also changed? Because if the orientation of pole changes, then the orientation of the spindle is also different. Now let us draw the chromosomes. One and two. We are starting with the cell which has undergone meiosis 1. So the chromosome number has reduced to half. What do we have to remember here? We have to remember that each chromosome, I'm going to draw a stage for you so that it is a reference point. This was the homologous pair. In anaphase, one chromosome went to one pole, other chromosome went to other pole. It went into one nucleus, this went into other nucleus. And each chromosome is with double DNA. So the chromosomes which have reached here, each chromosome has two sister chromatids. So now in this cell, how many chromosomes are visible? Only 2 per cell, because the daughter cells, they received half the number of chromosomes. Our parent cell, we started with 4, 2 pairs. This was one pair. Suppose this is the second pair, So this is one from this and one from this. 2 have reached one daughter cell and 2 have reached the second daughter cell. And each chromosome is with sister chromatids. So what has happened in prophase 2? Orientation of poles and spindle fibers is at right angles to the first. If first is vertical, second is going to be horizontal. If first is horizontal, the second is going to be vertical. But it will be always right angle to the first, unless and until the type of division is different. Like in periclinal division, the poles are going to be only one type. This we will take up later on, but I can give you a brief thing. Suppose this is one cell and cytokinesis takes place like this. That means one pole is here, the second pole is here. The cytokinesis was horizontal, the spindle was vertical. Suppose the same pole or same line of the pole is seen. So the first cell grows, the second cell grows. Here the poles of this cell and this, so cytokinesis will be like this. So as a result of it, you will get a cell, or rather cells, which are in the form of a long rod-like structure. If this is a cell and the division is cytokinesis is vertical, suppose the division is vertical, that means the poles were here, the two cells, they grow to their normal size, and again the poles are like this, so cytokinesis will be like this. So in this case you would get cells which are in the form of a sheet. So there are divisions where division takes place only in one plane. So poles are always from vertical, 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 or horizontal, horizontal, horizontal. But that sorry. But that is if you want only one type of structure to be formed. Say you want a membrane to be developed, or if you want a rod-like structure to be formed, then only one plane. But if you want mass of the cell to be formed, then in that case some cells should be inner, some cells should be on the sides. So in that case what happens is, suppose this is the cell and this is these are the poles. So cytokinesis takes place like this. These two cells grow to their normal size, and now the orientation of the pole or spindle is going to be at right angles to the first. In the first case the poles were horizontal. Suppose now the poles are vertical. So cytokinesis will take place like this. So there are cells which are on the outer side, there are cells which are on the inner side. So mass or a tissue can be formed. So normally, because this is the most common type, this is what we talk of. So if first is vertical, second is going to be horizontal. If first is horizontal, second is going to be vertical. So in meiosis 1, our poles were vertical. The spindle fibers were vertically formed. So in the second case, it is going to be horizontal. Second important point, spindle fibers, aster rays, assemble. Everything same, what takes place in any prophase change, be it simple prophase of mitosis, be it prophase 1 of meiosis 1, though it took place in the last one, that is dikinesis. But the changes are same. Membranes are going to dissociate, spindle fibers are going to assemble. So in both the cells now, the same thing has happened. Fibers are again two types. This one is continuous spindle fiber, and the one which is attached to the chromosome is chromosomal spindle fiber. Asters are also formed. This structure is the aster. And as we are talking about animal cell, you can see two asters in each cell. This is what is happening in both the cells which are in prophase 2. These cells have undergone interkinesis. Interkinesis is when the cells have prepared themselves for the next division. So in this particular part there is interkinesis is only preparation in terms of ATP, proteins, enzymes, etc. No DNA replication. Actually, DNA replication is not required because as we can see, each chromosome has double DNA. So now when the further steps take place, these two DNAs, that is original and the copy, they're going to get separated. Metaphase 2. Let us draw the cells and the basic structure which is still the same. And here we will draw two cells, two daughter cells. The centrioles were here because the orientation of the poles has changed. And then we also made the spindle fibers, aster rays. What is the characteristic feature of a metaphase? In any metaphase, be it metaphase 1, 2 or only metaphase, chromosomes have to align on the equatorial plane. There is going to be that intermediate phase where the chromosomal fibers are going to show differential contraction. Some are going to pull the chromosomes little up, some are going to pull them down, so that all of them, all the chromosomes come on the equatorial plane. So here this is the continuous fiber, here also that continuous fiber, and these are the fibers, the chromosomal fibers, which are going to get attached to the chromosomes, and the chromosomes, they have aligned on the equatorial plane. So let me draw the chromosome here. So this is one chromosome and this is the other chromosome. Two chromosomes in each cell. The fibers, these are the chromosomal fibers which are attached. So now what has happened here is, if this is a chromosome, each chromosome having sister chromatids is attached to two chromosomal spindle fibers, one from each pole. So here is a spindle fiber and here is a spindle fiber. Remember what happened in metaphase 1. Let me take you to that diagram which we made when we were trying to revise. Okay, this one. This is our metaphase 1. Here also the chromosomes, they came on the equatorial plane, but it was a pair of chromosome which was on the equatorial plane, and there is only one fiber which is attached to one chromosome and only one fiber which is attached to the other chromosome of the pair. In this case, what is happening is that there are two fibers which are attached to the same chromosome. So one is from one pole, other is from other pole, and in this case there will be pulling of the same chromosome from two ends, and this would result into splitting of the centromere. But let us first talk about the metaphase 2. Chromosomes reach the equatorial plane. What is attached to the chromosome? One spindle fiber, chromosomal spindle fiber from one pole is attached to one chromosome of, or rather just one chromosome. So only one chromosome, and the spindle fiber is attached. From one pole to the is attached to the chromosome from one end, and other spindle fiber from the other pole. So centromere of one chromosome is attached to two spindle fibers. One is coming from one pole, other is coming from other pole. So it is clearly visible from our diagram. Here there is no pairing. Single chromosomes are there on the equatorial plane, and from one pole one chromosomal fiber comes and attaches to the chromosome, and from the other pole other spindle fiber attaches to the same chromosome. This is metaphase II. Now after metaphase II, anaphase II. Here what we can do is we can draw the changes in one cell, because now we know what happens in one cell will happen in the second cell also. In this case, the spindle fibers will start to contract. So let us draw these centrioles here and the spindle fibers. Aster rays are still clearly visible. The continuous fibers are as it is, and the chromosomal fibers have contracted. So if the chromosomal fibers contract, what is going to happen to the chromosomes? So I'm going to draw that chromosome again here for you. This was the chromosome. Each chromosome with sister chromatids, and this is the kinetochore to which the spindle fiber is attached. So when these spindle fibers contract, it splits from here. The centromere splits. So this spindle fiber will take the chromosome with one copy of DNA, and this spindle fiber will take the chromosome with other copy of DNA. Centromere splits. Chromosomal spindle fibers contract, pulling the chromosomes, because of which what happens? The centromere splits, and whenever centromere splits, what is that structure that we see? We see a fiber, a new type of fiber. What do we call that fiber? Interzonal fibers appear between the split centromeres. So we will have to draw those interzonal fibers. Okay, we have not drawn the chromosome here. This chromosome is here. Okay, so the fiber that we have drawn now is interzonal fibers. And because it is an animal cell, in anaphase itself, we may start seeing little constriction in the plasma membrane. That is the beginning of cytokinesis. So little inward fold constriction may start. So in anaphase 2, what has happened is the chromosomes which were at the equatorial plane in metaphase have been pulled by two chromosomal spindle fibers from two poles, and this results in splitting of the centromere. Whenever centromere splits, there is interzonal fiber which appears. Don't you think it is same as what happened in anaphase? Anaphase, when I am saying anaphase, we are talking about mitosis. That means meiosis II, there are many things which are very much similar to mitosis. I did not say exactly same, I said similar. What is the difference? The orientation is different. So if orientation of poles is different, spindle is different, cytokinesis will also take place at a different plane. So there is slight difference, but it is pretty much same because centromere splits, interzonal fibers appear, and if interzonal fibers appear, then what are the forces which are going to take the chromosomes from equatorial plane to the poles? Three forces. One is contraction of chromosomal spindle fiber, then stretching of interzonal fibers, and third thing is ATP, because to move one chromosome from the equatorial plane to the pole, normally 30 ATPs are required, so ATP will be there. So these are the three forces which are going to take the chromosomes from equatorial plane to the poles. Same thing happened in the anaphase of mitosis. So that is why we say that meiosis II is similar to mitosis. So anaphase II has taken place. What will happen in telophase? In telophase, let us draw these two cells, and we can show that little constriction in plasma membrane which has appeared. Telophase is reverse of prophase. So everything that happened in prophase, just reverse of that, will take place in telophase. In prophase, or if you are talking about prophase 2, same thing. Nuclear membrane dissociated, here it will reassemble and appear. Nucleolus dissociated, here it will reappear. Chromatin fibers condensed, and you started seeing them as chromosomes. Here they will decondense and we will start seeing them as fibers. So let us draw the nuclear membrane which has reassembled towards the two poles. Aster rays, spindle fibers, they have now dissociated, so only the centriole will be visible here. Aster rays are gone, spindle fibers are gone, because aster rays and spindle fibers, they appear during prophase because assembly takes place. Chromosomes have decondensed, and because of this decondensation, fibers will be seen. How many chromosomes have reached the two poles? Two chromosomes are going towards two poles. So in this case, when we draw those chromosomes, but we will draw it as fiber. So 1 and 2 towards one pole, which is now enclosed in the nuclear membrane, 2 here, 2 and 2. So this is reverse of prophase. And after telophase, the cytoplasmic division will take place. So here what are the changes those are seen? Nuclear membrane nucleolus membrane of organelle reassemble and reappear. Reverse of prophase 2, asterase, spindle fibers dissociate, so they are not visible now. Chromosomes decondense, become thinner and appear like threads. And the constriction which has developed, that will get little deeper. This is what is telophase 2. After this, cytoplasmic division will take place. So here we will do one thing, we will draw cytokinesis in two parts, early cytokinesis and late, so that it is easier for us to understand. This is one cell and this constriction has become deeper. This is the second cell and its constriction which has become deeper from the other side. And when this constriction gets deeper and deeper, the cytoplasm will get completely divided and there would be 4 daughter cells. Okay, let us make the nucleus of this cell. Here also, when 4 daughter cells are formed, their centrioles are as it is. So let us make that centriole, and we are making it at the same position where the poles were. And inside each nucleus there are only 2 chromatin fibers, 1, 2 in each cell. So these are 4 haploid daughter cells. And how has this happened? It got completed in 2 parts. We started with a cell which had 4 chromosomes. This cell underwent meiosis I. We got 2 daughter cells having 2 chromosomes each. These 2 daughter cells underwent meiosis II, and we got total of 4 daughter cells each having 2 chromosomes. How was this possible? Because these 4 chromosomes, one pair and the second pair, this is how we got the 4. So, and let us make this as solid pair and this as the hollow pair. So when meiosis I takes place, one of the solid comes into this cell and one hollow comes here. One solid comes here and one hollow comes here. And then when this divides, because these 2 chromosomes, they have their sister chromatids, so this splits. So one solid in this, one solid in this, one solid in this, and one solid here. One hollow, just for our demarcation we are drawing it like this. So all of them, they have two chromosomes. So where was the DNA replication done? DNA replication was done before the cell entered meiosis, in interphase. That is S phase of interphase, where the DNA replicates. So when meiosis 1 takes place, chromosome number is reduced to half, but each chromosome is having double the amount of DNA. And in meiosis 2, that original and the copy DNA also separate by splitting of centromere, and that is how each daughter cell receives half the number of chromosome and the normal DNA content, that is according to that DNA. One DNA would have whatever number of or whatever C value of that genetic material. So this is how meiosis 2 is completed. It's a long process. Meiosis 1, prophase 1, longest. After prophase 1, metaphase 1, anaphase 1, telophase 1, cytokinesis may or may not take place. Then there is a short preparatory phase and then meiosis 2. In meiosis 2, same phases: prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinesis 2. So this gives us 4 daughter cells. Now what is the significance of this division? We know it is a reductional division. So when we talk about a reductional division, that means chromosome number is reduced to half. But the statement says that reductional division is essential for species specific diploidy. Imagine if this reductional division doesn't take place. So this is a cell with 46 chromosome. This is a cell with 46 chromosome. These two cells, when they fuse, we do not get the 46 chromosome species specific chromosome number. So this is going to be something else. So what happens is, organism, the parent, has 46 chromosomes. Meiosis takes place. And they produce 23 chromosome containing gamete. Now when these 23 chromosome containing gametes, they fuse, a zygote is formed with diploid number of chromosome. So this is our species specific diploidy. So in our case 2n is 46. This is species specific, human species specific chromosome number. And how are we able to achieve this species specific diploidy? Is because of meiosis. So meiosis is essential for attaining species specific diploidy. Very important point. It results in genetic variations. What exactly are genetic variations? Genetic variations are the differences which are seen in the genetic makeup in the members of a population. I'll give you an example to understand what and how this is going to help. In human population, we see people who have little darker skin, some people have intermediate skin color, some people have lighter skin color. This is variation. In human population, we see different skin colors. We also see different heights. Now, there is a variation. How is this variation achieved? It is due to crossing over. So due to crossing over, the genes which are responsible for more melanin formation, they are being passed on from generation to generation. And this is going to help in evolution, essential for evolution. How would it help in evolution? Suppose there are 3 or 4 different skin colors that exist. This is a variation. We do not get any benefit, we do not have any kind of a drawback because of that. But if the conditions change, which is actually happening now, ozone layer is becoming thinner and thinner. So if ultraviolet radiations penetrate into the Earth's atmosphere, what is that thing which can protect us? It is melanin. Melanin is our natural sunscreen. So if that change takes place, people who have more melanin or who have darker skin, they would have better chances of survival. And those who survive, they reproduce. And those are the ones which evolve. Because what is nature going to select? Nature is going to select which is the best suited for that condition. So there are variations which are going to take place, and variations are always because of crossing over. And those variations are very essential for evolution. So these are the two important significant points about meiosis. Now, when we talk of divisions, we also need to talk about the types, right, variations, as we did in case of mitosis also. In meiosis also there are three types. Gametic meiosis is seen in diploid organisms. That means the organism is 2N. In diploid organisms, the cells which undergo meiosis, suppose this is a cell which is capable of undergoing meiosis, then this cell is known as meiocyte. It undergoes meiosis to form haploid gametes. And from one meiocyte, normally there are 4 haploid cells formed, out of which all 4 or few would act as gametes. This you will understand when we come to spermatogenesis and oogenesis. This is seen in most of the higher animals. Zygotic meiosis is seen in case of haploid organisms. Suppose this is a haploid organism, this is also another haploid organism, and when they have to reproduce sexually, the haploid organism starts to act like a gamete. They fuse, a diploid zygote is formed, and the zygote undergoes meiosis to form 4 haploid cells, and these haploid cells live like organisms. So zygote is undergoing meiosis. That's why it is called zygotic meiosis. It is seen in lower plants like algae and bryophytes. The third is called sporogenetic meiosis. It is seen in plants like pteridophyte, gymnosperms and angiosperms. There are diploid spore mother cells. They can be called megaspore mother cells, microspore mother cells, and these diploid mother cells, microspore or megaspore mother cells, they undergo meiosis to form haploid spores. So it is like forming haploid gametes, but the spores are formed. And that is why it is called sporogenetic meiosis. So there are 3 different types of meiotic divisions. This is seen in case of higher animals, this is in case of higher plants, and in lower plants like algae, bryophytes, there is a different one, because these organisms are normally haploid. So those haploid organisms lead their life as haploid cells, but when they have to reproduce sexually, they themselves behave like gametes, and 2 haploid organisms will fuse. Zygote will be formed, and then zygote undergoes meiosis to form haploid cells again, and these haploid cells will again start leading their life as haploid organisms. So this is our meiotic division. And in the beginning of this lecture, I told you that the subscription plan, the pricing is going to increase very soon. So you decide as fast as you can to go for that subscription, be it 1 month subscription, 3, 12 or 24, depending upon your convenience and what you think is best for you. But try to do it as early as possible so that you get the advantage of the lower fee structure, because it is going to increase. And don't forget to use this code so that you are able to avail that 10% discount. Okay, so this is meiosis, mitosis, everything done. In the next lecture, we are going to start something new. And don't forget to watch the other channels also, where I'm going to start other chapters other than this channel. So hope to see you on all other channels very soon. Till then, take care and keep revising.